One of the goals of Diabetes A1c Forum is to inform diabetic patients of research that promises to improve diabetes care and the health outlook of patients at the molecular and cellular level. Today we're going to speak with a diabetes researcher named Dr. Denise Faustman about her work on a vaccine to reverse type 1 diabetes. Dr. Faustman is Director of Immunobiology at Massachusetts General Hospital and Associate Professor of Medicine at Harvard Medical School. Her type 1 diabetes research has earned her many awards, including the Oprah Achievement Award in 2005 for top health breakthrough by a female scientist, and the George and Judith Goldman Angel Award in 2011 for research to find an effective treatment for type 1 diabetes. As we speak, Dr. Faustman and her team are preparing to begin phase 2 human clinical trials with her vaccine, and will review her work beginning with laboratory mice, all the way through phase 1 human trials. She'll then discuss the goals and objectives for phase 2 human trials and tell us what additional steps are required to complete her work and hopefully make her vaccine available for doctors to prescribe. From Peninsula Television, this is Diabetes A1c Forum. Diabetes researcher Dr. Denise Faustman joins us today via Skype to talk about her work on a vaccine to reverse type 1 diabetes. Dr. Faustman, welcome to Diabetes A1c Forum. As a lead-in to our questions about your research, I'm always finding that it's important to differentiate between type 1 and type 2 diabetes. So I wanted to see if you could give a brief description from your perspective of what each of those is and how they're different from each other.
Type 1 diabetes used to be called juvenile onset diabetes, which means that it used to be thought that it was a disease of little kids. But since then, we've learned that type 1 diabetes can occur at any age. You can be 2 years old or you can be 40 years old. So its name got changed to type 1 diabetes. Type 1 diabetes is an autoimmune disease, which means that effectively you have bad white blood cells. And those bad white blood cells attack your pancreas and kill the insulin-secreting cells, and your blood sugar goes up. So a totally different disease from type 1 diabetes is type 2 diabetes. And in type 2 diabetes, you're usually an adult, and commonly you don't exercise too much, and commonly you're somewhat overweight. And in type 2 diabetes, the white blood cells aren't bad at all. And in fact, in type 2 diabetes, the problem is your pancreas just kind of wears out because you need a lot of insulin supply if you happen to be overweight. So although both patient populations use insulin, obviously, type 1 is a different disease than type 2. And type 1 is more similar to other diseases like multiple sclerosis, or psoriasis, or hypothyroidism. So it's in a different category of etiologies.
All right. Well, thanks for that explanation. Our viewers will really appreciate that and help them to understand the difference between the two better. So let's get into your research. And with regard to your work, what compelled you to begin research with diabetic lab mice back in 2000, 2001?
So the drive to do those experiments, which, believe it or not, are actually in humans now. We're heading into phase 2 trials in humans. But the reason we wanted to do the experiments was that we had worked a long time in the islet transplant field, trying to put cells in that made insulin. And we had discovered about 20 years ago that when you put the insulin-secreting cells in people with type 1 diabetes, the disease just reoccurs. So we knew there was a big, bad problem. And the big, bad problem was we needed to get rid of the autoimmunity, the bad white blood cells. So the reason we started the mouse experiments was to see if we finally could get islet cell transplantation going in mice that had the underlying autoimmunity that we thought we could take away.
All right. Well, so what did the research on the mice include? What kinds of things did you do in that research, and what did you learn from it?
Yeah. So we learned something pretty fascinating. So as we say in the business, this is as good as it gets. So and people out there in the lay field should realize that when you do these kind of studies, it's not like the data falls from the sky. It's a lot of hard work. And so we had worked about 10 years on just the bad white blood cell of people who had autoimmunity and mice that had autoimmunity. And we thought finally we had found the magic bullet to try to kill those bad T cells, those bad white blood cells. So the experiment was to set up to do islet transplants in these end-stage diabetic mice that had real autoimmunity, and put in the islet transplants and give them this drug to get rid of the autoimmunity. And there was a surprise. And the surprise that was totally unexpected was that the animals regenerated their pancreas.
Wow.
So instead of needing the islet transplant, we found out what we really just needed to do for type 1 diabetes is to get rid of the autoreactive cells, and that there was spontaneous regeneration.
Wow. Wow, that's
So
great.
that was the big surprise.
Yeah, I'll bet. So if we fast forward to 2008, when you began phase 1 human clinical trials, during the period between when you completed the research with the mice back in 2001 and 2008, how did you determine it was worth the risk to go from experimenting with mice to investing the time and expense to human clinical trials?
Yeah, that's actually a great question, because even people in my own field can make that mistake. You know, you can have great looking mouse data, but it has no applicability to humans with the disease. So what we did in that time period was actually intensely study people that had type 1 diabetes and all stages of type 1 diabetes, because we wanted them to come into the clinic. We wanted to draw their blood. We wanted to look at their blood and see if there was any evidence of these same bad autoreactive cells. And not only did they have the bad autoreactive cells, but could we put the drug in culture on the cells and similarly kill the cells causing the disease? So that's what we did as a segue between the mice and the humans. In other words, we wanted, before we just got all excited and moved things forward into humans with a lot of work, to make sure humans had similar white blood cells that could also die with this therapy.
Well, you know, that brings home a point that I wanted to get across in this interview, and that is most people like myself are not familiar with the type of work that you have to do, and I really want to drive home the amount of hard work that it takes to get to each one of these steps. And I'm sure that you had to, even after doing that, you had to convince a lot of people and get a lot of the people with the right skills involved in order to get going with phase 1 trials.
Yeah, I mean, it's absolutely true. It's not only skills, but it's also money. Okay.
Yeah.
Okay. It's skills at raising money, right?
Yeah.
So, especially when you start out doing experiments with data that's maybe going to change the paradigm, and we were changing two major paradigms in this disease.
Yeah.
One was the fact that these mice, for the first time, were end-stage diabetic mice, and we thought that maybe, although it was a really lofty goal, what we should really be doing is developing immune interventions for people that had the disease, not just people who had new onset diabetes. So that was a real paradigm change, because if you look through all the trials in the world that are still going on, they're trying to treat people that just got diabetes yesterday or a week ago or 4 weeks ago or a month ago. We decided to take the really big step and start to think about immune intervention trials in people with the disease. So that was a paradigm changer. And the other paradigm changer was that we thought we had really identified a drug, a compound, and even a cheap way of trying to remove the disease. So we were changing a lot of paradigms, and clinical trials are expensive. Mice are cheap in comparison, but we knew the data, and that's why we wanted to take the big step forward.
Yeah, and that's those two paradigms you're talking about here are what really got me interested quickly in the work that you're doing. When you decided to go ahead with phase 1 human clinical trials, what were the goals that you set out to accomplish for phase 1 trials?
Yeah, so for phase 1, we had some goals that maybe the public wouldn't necessarily appreciate right away, but we thought were really important. And we spent that time period trying to do what we call biomarker development. And what that means is that you wouldn't administer insulin unless you could check a blood sugar, right?
That's correct, yeah.
It just doesn't make sense, right?
Yeah.
The chance of you being able to dose insulin without a blood sugar is about zero. You'd kill some people and a lot of people would die. And we thought that as we move forward into human clinical trials, it was critical, absolutely critical, that we develop the blood test for humans so we could see if they had the bad T cells, and even with low dosing of this drug, that those bad T cells started to die. So that was one of our goals. Could we validate these markers, these blood markers, to show the drug was working at early stages of dosing? And the other goal was to develop very meticulous tests to look for low levels of pancreas function. So we were, to us, it was as important doing the basic science behind these trials, and not just march into people hoping you were lucky that day and got the right dose at the right time.
So you're kind of building a foundation there, a credible foundation that you could work from by doing that work.
Yeah, and keep in mind that, people should be skeptical about trials. I mean, if you look at the trials the last 5 to 8 years in the United States and in the world in type 1 diabetes, most of them haven't worked, right? We're talking billions of dollars in trials. And that's pretty depressing. And you go back, and of course, hindsight's always really keen, right? But you go back and you go, well, there's great excitement in mouse, but did anybody ever look to see if humans had the same defect before you went into humans? So we wanted to be cautious, but yet we really wanted to do the tough science of trying to do new things. So we tried to pair it with the basic science as we went into phase 1.
Oh, that's great. So what is, can you explain what the vaccine is based on that you're working with, and what it does, and are there any side effects to it that you found yet?
So the drug we're working with most is called the BCG vaccine. And this vaccine, if you look in Wikipedia, has been around for over 90 years, almost 100 years now. And it was originally developed in the world and is still used today. It's the most common vaccine administered in the world today as a way to try to prevent tuberculosis. Now, we're obviously not using it to prevent tuberculosis, okay? We're using it for a different reason. And the different reason we're using it is the fact that it's known that when anybody gets this vaccine, you release a hormone called TNF. And it's really TNF we wanted to go in trials with, but TNF's not available. It's not manufactured. You can't find it in Argentina. You can't buy it in Mexico. You can't buy it in Russia. Believe me, all the diabetics have been on the internet. I know you guys looking for TNF, and it's just not manufactured. And so we decided that since these were nonprofit trials, supported totally by philanthropy, that we would try to give back to the public by developing an inexpensive way to move forward. And that was to reuse this vaccine that elevated TNF. So it was really the elevation of TNF we were trying to go for.
Okay, so are there any side effects with BCG or raising TNF in the body?
Sure. So when you have about 4 billion people that have been vaccinated with BCG, you have an amazing base of safety data, unlike new drug development or even something that's a prescription in your pharmacy. So we know a tremendous amount about what happens when you get the BCG vaccine. So like any live attenuated vaccine, like the flu vaccine, you shouldn't get the BCG vaccine if you have AIDS, and you shouldn't get the BCG vaccine if you're on immunosuppressive drugs, or if you're undergoing chemotherapy for cancer. So you never want to pair it with a drug or an infectious disease condition that suppressed your immune response. So that's one counterindication. Or if you have like one of these rare genetic diseases where you don't have white blood cells, like the bubble baby, you would never give the bubble baby.
Yep.
A BCG vaccine. But other than that, those are the standard counterindications. Oh, one more counterindication. Somebody in your household is immunosuppressed. So your mom has breast cancer and is getting high-dose chemotherapy. You wouldn't want to get the vaccine. So it's kind of the same list you see when you go to get your flu vaccine, same kind of questions of counterindications to taking it.
Oh, interesting. Okay. So I've known some diabetics who have gone and had a pancreas transplant to begin producing insulin again. Wouldn't that enable a type 1 diabetic to start producing insulin again?
Yeah, so another technology that, was really, we worked on pancreas transplantation and more so islet transplantation for many years. And we realized that, well, you've got the hurdles of there's not enough islet supply, right?
Mm-hmm.
But the other hurdle is when you get an islet transplant or pancreas transplant, you need to go on drugs that are immunosuppressive drugs. So those are nasty drugs. Those are not drugs that, you casually go on. Now, if you need a kidney transplant, and kidney transplants are life-saving, of course you're going to go on immunosuppressive drugs. It makes life without a kidney is not a good life. So those drugs look good. But if you're 40 years old and you're taking insulin, going on those drugs doesn't look so good with respect to the drug side effects.
Mm-hmm.
So we're trying to develop something that has an impeccable safety record and is not an immunosuppressive drug to treat people who have long-term diabetes.
All right, that's good to know. So with regard to BCG, since it's commonly available, why wouldn't doctors just start prescribing that now to patients that they have that are diabetic?
Well, that's always a tough one. So the way I put that is, you can imagine this phone right over here, you can't see, about once a month, I get a phone call. Usually it's, Dr. Faustman, we need a confidential call, and it's Dr. So-and-so from someplace. And they tell me in detail that their son or daughter's diabetic, and they want to know what dose to use of BCG. And I'm always kind of amazed that people would think that I would know the dose but wouldn't share it with everybody. And so it's kind of like any new thing. Would you dose? This isn't totally analogous, but it helps you understand. Would you randomly dose your kid with insulin without a blood sugar?
No.
Now, if they were dying, you would. But if they're not dying and they have a standard of care, you'd say, call me when you get the right dose.
Yeah.
So I think the beauty of what we do is that we think what we can do is work in all different stages of diabetes. And unfortunately, a lot of the diabetes community's been very sensitized that if you don't get in a trial when you have new onset, then you're off the list for the rest of your life. And we keep reminding people that these trials are truly unique and we're treating people with the disease, not just new onset kids. So they have a little window to wait so we get the dosing right. So that's the best explanation we can give for wait till we get it right instead of dosing incorrectly.
Okay, that sounds good. So, is there going to be a standard dose for people diabetics across the board, or are you finding that that's got to depend on the patient?
Yeah, well, we're going to find out more about that in phase 2, but the dosing we're talking about is not like insulin dosing. So we're not talking about giving yourself 6 injections a day and checking your blood sugar 6 times a day. We're talking about whether would the dose would be once a month, once a year, once every 5 years. So we're talking about a whole different scheme of medical intervention compared to insulin.
Okay. So good lead into phase 2. When are phase 2 human clinical trials set to start, and what are the goals for phase 2?
Okay, so for phase 2, since we had success in phase 1, early signs of success in phase 1, you don't want to change too many things when you go into phase 2, okay? So phase 1 was remarkable in that the people were 15 to 20 years out from type 1 diabetes. Phase 1 was remarkable in that in very low dosing, we could start to see the early signs of insulin return from the pancreas. And we could also see the death, but there was still a burden of bad T cells, the death of the bad T cells. So the first group that we'll start with in phase 2 will be people, again, with longstanding type 1 diabetes. We're doing lots of screening right now. So we always ask everybody, here's my sales pitch, to log into our website, which is faustmanlab.org. So it's FAUSTMAN.org. And there's registration sites there. So you can register. And then you can follow it up and come to Boston and get your blood drawn. Or you can just register and say, call me if I look interesting to you. Or you can go for a weekend vacation in Boston and stop by on a Monday, our favorite visit day. No coincidence. And get your blood drawn. Or you can come on a Friday and then spend the weekend in Boston. So we're lucky to be in a popular city.
Yeah.
And people sometimes on business travel just stop. You have to make appointments ahead because we're really pretty booked. But everybody can participate, and it may just be donating a tube of blood, or it may actually be within one of the recruited groups. And we'll be recruiting different groups over the next 3-4 year period. So it pays to get registered on our website and get updates, and we'll keep you in the loop.
All right. So in phase 2 trials, you're just going to continue what you were doing in phase 1 and see if you can get
more prolonged effect, yeah. So
Yeah.
we're going to be giving more vaccines. So
Okay.
in phase 1, we gave 2 small doses of vaccine 4 weeks apart. And in phase 2, we're going to be giving vaccines out for 5 years. So many more vaccines to try to get the pancreas to regenerate and hopefully to start to correct the hemoglobin A1cs.
Yeah, well, you know, I wish you very much luck with that because of my own case. So what additional steps beyond phase 2 human clinical trials have to occur before a BCG vaccine becomes available for doctors to prescribe?
Yeah, so obviously these trials, even though we're doing them within academia, are going not only through full, Harvard MGH institutional approval, but they're going through the FDA for approval as well. So it takes us a little longer, but then the data is very well respected because of the strict oversight that occurs. We're actually developing BCG for what we call licensure, so that it's not an academic experiment where we tell you in 5 years we got great data, got a great drug, and guess what? You can't have it.
Heard that before.
Yeah, but it's true. I mean, it's faster and easier to do it that way. But we're very fortunate, since it's a cheap vaccine, that we're moving forward, that the public and foundations have come through with support to try to really change type 1 diabetes care by this changed paradigm. And so because of that generosity of the public, we feel it's really our obligation that if we can show efficacy, to bring it forward all the way to something that your doctor can prescribe, and the safety of the drug is well verified by the FDA. So in general, phase 2 trials, as I mentioned earlier, we're starting with one type of clinical type 1 diabetic patient. We'll also start enrolling other types of patients as we move through with longstanding diabetes, and we'll see what the regulatory bodies tell us as we move through. The advantage of working with 90 to 100-year-old drug is that the FDA likes drugs that have 100 years worth of safety information in them. So we're hoping we can continue to move at this fast pace to get it to actual endocrinologists being able to prescribe it.
Okay, so when you, one other question I have going back to the killing these rogue T cells or bad white blood cells, how do you avoid harming other healthy cells in the body?
That's an excellent question. So it's actually kind of a deep science question, so you get major kudos for asking me this one.
Thank you.
So it turns out that it's kind of the analogy I can use for the public here is it's kind of like an antibiotic. And most people, most days, when they take an antibiotic, they don't die from an antibiotic, right?
Yeah.
And you go like, why don't you die? Why do the bacteria die, but I don't die? And the reason, if you read like, a high school biology book, is that the cell wall of the bacteria is different than your cell wall. So the antibiotic interferes with cell wall synthesis, generally, some types of antibiotics. And so when you take the antibiotic, you don't die, but the bacteria die. So what we found, okay, and this is not like a lightning bolt and we got the data, but what we found by studying human lymphocytes from lots and lots of volunteers is that we found a membrane difference between the diabetic and the normal and the diabetic bad T cells and the normal T cells. So how BCG works is that it makes this hormone in your body called TNF. And it's actually this hormone, also known as a cytokine, that only kills the bad T cells. So it's a very targeted way to attempt to reduce the disease burden. It is not an immunosuppressive drug where the immunosuppressive drug kills all the T cells or inactivates all the T cells. This drug, at the right dosing, should only kill the cells that are killing the pancreas.
Okay, so do we know, Dr. Faustman, how these cells become rogue or bad, or is...
Yeah, that was part of the basic science work we started out on. Whether they are generated in your bone marrow, your thymus, it turns out that Mother Nature's a little bit inefficient, and everybody makes millions of T cells every day of your life. And unfortunately, most of those T cells, or fortunately, most of those T cells never get out of your bone marrow thymus because they're autoreactive. And so there's a very strict teacher cell in the bone marrow and thymus that negatively selects those Rug T cells, and they should never get out of your bone marrow. But what we know in diabetes, as well as other autoimmune diseases, is that though that teacher process is interrupted, and that teacher is not a very good teacher, and as a result, those Rug T cells get out. And the beauty of TNF is it can then find those Rug T cells and kill them at a later time in life.
All right. Well, Dr. Faustman, unfortunately, we're out of time. Very interesting topic. Want to thank you for taking the time to inform us of your research on today's show.
Thank you for inviting me.
All right. Take care. This episode of Diabetes A1c Forum is intended to expand your perspective on diabetes research and introduce you to the real capabilities of researchers like Dr. Faustman. Her work is particularly significant because of two major factors. One, it focuses at the molecular and cellular level where the disease occurs. And two, the goal is to create a vaccine with the potential to measurably improve the health outlook of type 1 diabetics. There's a lot of diabetes research being conducted, but we don't often hear about it or what the results were. Stay involved because Diabetes A1c Forum will periodically host other researchers making valuable contributions that should be highlighted and promoted. Join my blog at www.diabetes-a1cforum.org and rate this episode on a scale of 1 to 5, 1 being excellent and 5 being terrible. This will help me to determine what future shows on diabetes research should include. Thanks for watching, and we'll see you next time on Diabetes A1c Forum.